In this video, we are going to have a short tutorial on how glycogen is metabolized in the body. Don't forget to like and subscribe to the channel after watching so that you will receive every update from this channel. Glycogen is a homopolysaccharide which is made of several residues of glucose. It is stored in the liver when the body's starch level or when the body's glucose level tends to be in excess. Its metabolism involves either its synthesis or its breakdown. Now, its synthesis by the process of glycogenesis occurs when the glucose level in the blood tends to be excess, and its breakdown is by glycogenolysis when the glucose level occurs to be limited. Now, glycogenesis, which involves the synthesis of glycogen, is controlled by a hormone released by the pancreas we call insulin. This insulin is released in response to excess glucose in the body, and it signals the activation of the glycogen synthase, which is the main enzyme for the synthesis of glycogen. The breakdown of glycogen is signaled by hormone glucagon or epinephrine released also by the pancreas, which also signals the activity of glycogen phosphorylase, the main enzyme involved in the breakdown of glycogen to free glucose in the body when the glucose level in the blood is limited. The glycogen structure is made of several monomers of glucose linked together by alpha-1,4 glycosidic bond and branched by alpha-1 cis glycosidic bond. This means that for glycogen to be broken down, the branched bonds or the branched chain must be broken down by the branching enzymes to release all of them in a straight chain or in alpha-1,4 glycosidic linkage form so that they can easily be hydrolyzed to free the free glucose in the body. Now, this is a summary of the schematic representation of glycogen metabolism in the body. The metabolism of glycogen is signaled both by enzymes and hormones. When there's high glucose in the blood, the body tends to release insulin to signal the activity or to stimulate glycogen synthase. In other words, when there's low glucose in the blood, the body tends to release glucagon and epinephrine, which signals the activity or activate glycogen phosphorylase to break down glycogen. And when there is high glucose level in the blood, the body tends to store some as glycogen. So the first step in glycogen synthesis is the phosphorylation of the glucose level or the glucose residues in the blood by an enzyme glucokinase. This enzyme occurs in the liver, but it has its isoenzyme in the other cells like the cells of the muscle and that of the kidney called hexokinase. The hexokinase tends to have a higher affinity for the glucose than the glucokinase. This means that even in a limited amount of glucose level, the hexokinase can still phosphorylate the glucose to form glucose 6-phosphate, but the glucokinase cannot. When glucose 6-phosphate is formed by the glucokinase through phosphorylation of the glucose, an enzyme called phosphoglucomutase, a mutation enzyme, causes the movement of the phosphate group on the 6th carbon of the glucose to the 1st carbon of the glucose to form glucose 1-phosphate. That is when the body's glucose level tends to be excess. It is phosphorylated to form glucose 6-phosphate, then it is mutated to form glucose 1-phosphate. Now at this point, the glucose tends to be activated. An enzyme which is called the UDP glucose pyrophosphorylase catalyzes the depyrophosphorylation of UTP, uridine triphosphate, to form UMP. So UTP is made of 3 phosphate groups and 2 phosphates are being cleaved off to form UMP by an enzyme UMP glucose or UDP glucose pyrophosphorylase. Now the net UMP, which is made of only one phosphate, will bond to the phosphate group on the 6th carbon of the glucose 1-phosphate to form UDP. It is uridine diphosphate because it is di 2-phosphate, 1-phosphate from this one, the glucose 1-phosphate, and the other phosphate from UMP, which is cleaved or which is formed by the cleavage of the pyrophosphate from the UTP. Now, when the UDP is added to the UMP is added to the glucose 1-phosphate to form the UDP, this one is the activated form of the glucose, which makes it more reactive. Here, the glycogen synthase, which is activated by the insulin, will come and attach the glucose component of the UDP to the growing glycogen primer by cleaving the UDP off. So at this point, the glucose is activated, and is being attached to the growing glycogen primer after the cleavage of the UDP. This UDP is on the 1st carbon of the glucose to extend the glowing glycogen chain. So that's how basically glycogen is synthesized. It is first by phosphorylation to form glucose 6-phosphate, then mutation to form glucose 1-phosphate, activation to form UDP glucose, then cleavage and attachment of the glucose residue to the glycogen primer to form the glycogen. Now, when the glucose level in the blood tends to drop, this enzyme is inhibited by the activation of or the release of these enzymes. These hormones. So glucagon is released by the pancreatic cells as well as the epinephrine. These two hormones tend to activate the enzyme glycogen phosphorylase, which is the main enzyme in glycogen breakdown. Now the glycogen chain will first be phosphorylated by this enzyme at the carbon 1, then hydrolyzed. So the glycogen will be phosphorylated and hydrolyzed to form glucose 1-phosphate as the first product. The whole of this reaction is catalyzed by glycogen phosphorylase. And this one becomes activated when glucagon, epinephrine are released by the pancreatic cells, or several residues of adenosine monophosphate is released or occurs in the body. The same process, the glucose 1-phosphate, which is in need of the body needs it, will undergo mutation by the phosphoglucosamidase to form glucose 6-phosphate. So this glucose 6-phosphate is coming from the glycogen. After hydrolysis by the glycogen phosphorylase and cleavage to form glucose 1-phosphate, it is mutated by phosphoglucosamidase to form glucose 6-phosphate. And when we have this, the glucose 6-phosphate is an intermediate in glycolysis. If this reaction tends to occur in the muscle, these intermediates would enter the glycolytic pathway to produce pyruvate. The glucose 6-phosphate enters the glycolytic pathway to produce pyruvate, that is for energy generation, which later produce carbon dioxide in the Krebs cycle and water in the electron transport chain system, that is in aerobic condition. In anaerobic condition, the pyruvate goes into conversion into lactic acid. If this process occurs in the liver has an enzyme we call glucose 6-phosphatase. This is an intermediate enzyme in gluconeogenesis, and this enzyme cleaves off the glucose from the 6th carbon, the phosphate from the 6th carbon, to form a stable glucose unit. In the muscle, this enzyme is not present in the muscle. This enzyme is lacked in the muscle. So when you get the glucose 6-phosphate, it enters the glycolytic pathway for energy generation, for ATP synthesis. But in the liver, this enzyme is present. So this one tends to enter the gluconeogenesis pathway, and this enzyme cleaves off the phosphate on the 6th carbon of the glucose to produce the glucose unit, so that the body can use it. Let's say the brain can use it for its activities. So this is basically a summary of how gluconeogenesis occurs. If glucose is in excess, insulin is released, and this proceeds the gluconeogenesis or glycogenesis, this one, by activation of glycogen synthase. If glucose level is small or limited, the glycogen is broken down by an enzyme, glycogen phosphorylase, when glucagon, epinephrine are released by the pancreas, or when the body has lot of adenosine monophosphate in the muscle, which tends to also activate glycogen phosphorylase. Thanks for watching. Don't forget to subscribe.